Okay, it's a great pleasure to listen to Denis Pe. We heard from Pierre Sabatier about repairing the brain using very sophisticated molecular tools to engineer cells and do all sorts of things. And maybe there's another way of doing things in terms of using the brain to control itself. And this is, I think, what Denis is going to be talking about.
Thank you, Henry. So thank you for staying for this second talk. I will take this opportunity to introduce very quickly what we are doing in the impact team. We are essentially working on the interaction between perception and action, because in most of our daily activities, we produce goal-directed acts to communicate and to interact with our environment. And when changes in the environment or in our own body are not corrected by the brain, by plasticity mechanisms that are deficits in sensory motor performance, and to the extreme position in the clinical side, action deficits cause severe handicaps. So it's very important to understand this coupling between perception and action. And the two main goals of our team is to identify mechanisms and also threat of goal-directed acts on the associated spatial perception and cognitive functions, and also in parallel to develop and test protocols of functional expression and rehabilitation of these different functions. So we are really interested in the bi-directional interaction between perception and action. So in the classical direction, we try to understand how the different sensory inputs are integrated to produce central representation and to go until the movement execution, action execution. And we are mostly interested in upper limb movement, eye and gaze movements, and motor control of the lower limb to control posture. And in the other direction, these movements produce feedback signals, essentially visual and somesthetic feedbacks, that can modify the action online. This is what we call the flexibility mechanism, or can train by training or by adaptation, can modify subsequent actions when we made systematic errors, this is called plasticity. And all of those longer term, we are also, of course, interested in knowing how the brain can change itself over the long term for rehabilitation or to compensate for brain lesions. So now I will turn to my own topic of saccadic eye movement. So why studying saccadic adaptation? So first of all, eye movements are a very good model to investigate action perception interactions. And they have also, on the clinical side, high diagnosis values. This is an example, for example, here of a cerebral patients. You see here the trace, the red trace indicates the eye position of this individual attempting to look at this target when jumping from the center to the right side. And this patient is making two large movements, hypermetric saccades, and making oscillation around the saccades, and which is quite typical of these cerebral lesions. And so also adaptation of saccadic eye movement is a nice tool to investigate sensory motor adaptation in general, and especially because saccadic eye movements cannot be controlled during their execution by sensory feedback. So in order to produce accurate saccade, we really need to calibrate precisely the motor commands sent to the methods. And also, so in addition to the potential to study brain plasticity mechanism, saccadic adaptation also is a very nice tool to dissociate perception from action. And hopefully it will also provide some new solutions to take care of patients with visual attentional deficits. So what is saccadic adaptation? Saccadic adaptation is a process by which we can maintain the accuracy of saccades over the lifetime, despite numerous changes in our organisms due to growth or to aging. And it can also compensate over a shorter term for central or peripheral lesion, as shown initially by these people in human subjects, and then by Optican and Robinson in monkey, that they operated, they weakened the muscles of one eye, and they observed, of course, that this surgery produced very inaccurate saccades produced by this eye. And then by patching the good eye, several days after, they noticed that the eye saccade amplitude had recovered. And this was produced by an increase of the innervation to the muscles, as shown by the increase of the size of the saccade of the other eye. So suggesting that some internal adaptation had occurred during this time. And I will not come back again to this point because I already said that. Just to mention en passant that Optican and Robinson also did the same experiment in monkey after cerebral lesion, and they noticed that there was no longer any adaptation left in these animals. So hopefully we have a nice, although very old, protocol that we can use to elicit adaptation in the lab non-invasively without making any surgery in our subject. So this protocol is what we call the target double-step protocol, because the target steps once, and the subject is making an automatic saccade toward this target, what we call a reactive saccades. And during the saccade, we measure the eye movement online, and we trigger a second step of the target during the saccade, so that the subject is unaware, does not notice this perturbation, as long as this perturbation, the size of this perturbation is not too large. But if you step the target back in this way, at the end of the primary saccade, there is an error, and this error is used for two corrections: an immediate correction, which is the corrective saccades, and then when you repeat this kind of error over some tens of or hundreds of trials, there is a triggering of the adaptation processes. So this repeating this error induces a progressive reduction of the primary saccade, which is the effect of the adaptation. So this goes like this. You present a visual target. When the saccade is launched, the target is jumping back. And you see that this procedure can decrease the size of saccades, and this effect is maintained after the adaptation phase, even though in this post-adaptation phase, the target is no longer jumping, which indicates that this effect is not due to some strategy, but it's really the result of a modification of the saccadic system. And we designed some variation of these initial protocols in order to study the adaptation of scanning voluntary saccades. These are saccades that we normally produce very often in our daily life to explore our visual environment. In the lab, this environment is very poor, but the principle is the same. We also are subject to make saccades at their own pace between visible targets without any sudden jump of the target. But during the saccade, all the display is jumping backward in this example, for example. And now I will try to summarize some years of research using these two paradigms in order to compare adaptation of reactive saccade and of voluntary saccade. So the first experiment that we did in this line was performed by Nadia Layan. With Nadia, we adapted reactive saccades and we measured the size of voluntary saccades. And we could measure in this case the transfer from reactive saccade adaptation to voluntary saccade. And we noticed that the transfer was very small. And in the with the same subject tested in a different day, we did the opposite. We adapted voluntary saccade and measured a very big transfer to reactive saccades. So this asymmetrical transfer between reactive saccades and voluntary saccade adaptations led us to suggest that already the saccade type has some importance regarding the locus of adaptation. And we propose that for reactive saccades and for voluntary saccades, we have different sites involved. Very similar sites in the downstream motor pathway. But for voluntary saccade, we suppose that there was an additional site which is located more centrally, more distal from the output, from the motor output. And this kind of a diversion was reproduced in a different lab, in a Paris lab, at about the same time. The second experiment in which we tested transfer was an experiment we did in collaboration with people from Marseille, Alain Guillaume and Julien Coty. And in this case, what we did was to test pointing movement, hand pointing movement, before and after saccadic adaptation. And when we do that, when we measure the change of size of hand pointing after reactive saccade adaptation, there is really no effect. But when we do voluntary saccade adaptation, there is a significant and strong transfer to hand pointing movement in the same subjects, again tested in a different day, suggesting that when we adapt reactive saccade, we really are dealing with motor pathway which is specific to the saccadic system and without any interference with the hand pointing movements. In contrast, for voluntary saccade, we are assuming that since there is a partial transfer to hand pointing, that there are again two locus of adaptation. This is mine. We have two sites but we have two sites for voluntary saccade adaptation, one which is very similar as for reactive saccade and one which is more upstream. So upstream from the site, so the upstream site would modify the signal sending to the hand pointing movements. And in the same spirit, the third study what we performed was to test the transfer of adaptation to anti-saccade. So why anti-saccade? In the anti-saccade task, subject is presented the visual target, let's say on the right, and has to respond to the opposite side, towards its mirror location. So this task allows us to dissociate a visual vector pointing towards the target from a motor vector, which is pointing in the direction of the movement, which is performed by the subject. And this operation is called vector inversion, which has to be done during the planning of an anti-saccade. And so we asked whether saccadic adaptation would be taking place downstream or upstream of this vector inversion. And to do that, we again adapted reactive saccades and tested the two types of anti-saccades after and before adaptation. So the adaptation of reactive saccade is shown by this significant decrease of gain. And when we looked at the anti-saccade directed to the right, after having adapted rightward reactive saccades, we observe a full transfer. So the change was the same as for reactive saccades. And there was no transfer to the other anti-saccades. So it seems that when the vector, when the anti-saccade, the anti-saccade which is modified is the one for which the motor vector corresponds to the adapted vector. But when the sensory vector of the anti-saccade corresponds to the adapted vector, there is no modification of this anti-saccade. So suggesting that the adaptation locus for reactive saccade is taking place below the level where this inversion of vector takes place. And in contrast, again, we found a difference if we adapt voluntary saccade, because in that case, we observe transfer to both types of anti-saccade, suggesting again that there would be two sites of adaptations for voluntary saccade. Again, one site located downstream and the other site located upstream from this level of vector inversion. So to conclude this first part of dealing with the different transfer studies, from independent studies, we came up to the same conclusion that for reactive saccade, it seems that there is only, we could evidence only, demonstrate only one site of adaptation, quite downstream. But for voluntary saccade, it seems that in each case we have two sites, one which is close to the sites involved for reactive saccade adaptation, and the one which is more upstream. So I will base my, the rest of my talk on these results, explain the hypothesis. But just before the hypothesis, the neurophysiological hypothesis, I will just mention that as for other types of motor adaptation, the cerebellum is really important for saccadic adaptation. I already mentioned that when I talked about Optican and Robinson data in the monkey. This has been confirmed by other authors, and also Michel Demerger in humans showed in a PET experiment that the medial part of the cerebellum, what we call the vermis, is activated during an adaptation protocol. And here are the lesions that these people made in the monkey cerebellum to produce a deficit of saccadic adaptation. So very similar to the site shown by Michel. So I will present an hypothesis that I proposed 2 years ago, based on these findings, on other findings of the literature, in which I will represent the sites involved for reactive saccade adaptation in red and for voluntary saccade adaptation in blue. So we propose that at the level of the cerebellum, there are overlapping neuronal population involved in these two types of adaptations, and the effect of this cerebellum will be at the level of the brainstem of the superior calculus or reticular formation, which are involved in producing all types of saccades. But in addition, based on the findings that I just mentioned to you, we propose also that at the level of the cerebral cortex, maybe at the level of the input to the parietal cortex, we suppose that some changes related to adaptation of voluntary saccades. So now I will present you a series of experiments that were aimed mostly at testing this kind of model. So first of all, we use TMS, transcranial magnetic stimulation, over the cerebellar hemisphere in healthy subjects with Muriel Panouillère in collaboration with Bas Negers and Martin Frans in the Netherlands. And to demonstrate that when we applied single pulse TMS on the lateral cerebellum, we can induce impairment of saccadic adaptation. So this is the first study showing that cerebellar areas outside of the medial part of the vermis could be involved in saccadic adaptation. We also analyze the behavior of cerebellar patients. Again, it's an experiment, a study performed by Muriel Panouillère and others. And we came up with the conclusion, again, this time by comparing reactive saccade adaptation versus voluntary saccade adaptation, that there are different types of deficits, suggesting that this anterior lobe, the hemispheric part of this anterior cerebellum, would be more specifically involved in saccadic adaptation. And also to our surprise that this posterior lobe would not be apparently crucial for adaptation, whereas the pathway, the cerebellar input coming from the medulla, when interrupted by lesions in Wallenberg syndrome patients, would interfere with both types of adaptations. So I go quickly just to have time to illustrate a little bit more the last two studies that we performed. The first one is an fMRI study that we did with Peggy Jardin, and the second one is again a TMS study that we performed with Muriel Panouillère on the parietal cortex. So I will detail these two studies now during the rest of my talk. So this study, the fMRI study, has been done in the SURMUP using the 1.5T system with the iScan eye tracker. So we had, because of course we had to monitor precisely eye movements while scanning our subjects. So we had 6 subjects tested either in a reactive saccade adaptation task or in a voluntary saccade adaptation task in 2 different sessions, separated by at least a week. And we analyzed the data coming from these scans with multivoxel pattern analysis. And to do that, we also had our subject making regular saccade pro or anti-saccade toward visual or memorized targets on both sides in order to identify region of interest, regions which are involved in visuo-oculomotor responses. And for each identified region of interest, we then use multivoxel pattern analysis to identify which one would be related to adaptation of reactive saccade and which one would be related to adaptation of voluntary saccades. For each of these 2 reactive saccades and voluntary saccade adaptation scans, we had 4 conditions. The 2 main conditions were adaptation and control. The adaptation condition is very similar to what I showed you so far. That is to say, a double-step target paradigm. The second step of the target, as compared to previous experiments, was triggered only 50 milliseconds after the main saccade. And the control condition, we did exactly the same thing, but this delay was lengthened until 500 milliseconds. Why did we use this delay? It's because some people like Fujita previously demonstrated that when you step the target from T1 to T2, some time after the saccade, you elicit corrected saccade, but you don't elicit any adaptation. So for us, this was a very good control for visual stimulation eye movements. And by comparing directly these 2 conditions, we hoped that we would be able to demonstrate activity specifically related to adaptation. And we had also two single-step also conditions that I will not present today. So first of all, I will present the network which is activated by the saccade localizer task. So this is a very large network at the level of the cerebral cortex, and in the cerebellum also, in the vermis and the lateral hemisphere, with many visual and visuomotor areas. I have to mention here that these regions of interest were computed by making the subtraction between saccadic movement and fixation. So we have here this activity is related to visual processing and saccade programming. And then we studied separately each of these regions of interest using MVPA analysis in order to know whether this region of interest would be able to statistically discriminate between our 2 main tasks, meaning that if yes, that would be an indication that this region of interest is involved in saccadic adaptation. And for reactive saccade, we found a pretty small number of regions of interest, 4 actually, 1 in the cerebellum and 3 in the cerebral cortex. 1 in the frontal cortex and 1 in the TPJ area and 1 in the MT area. And for voluntary saccade, in contrast, we found different areas, or we found also the cerebellum and the frontal area, but in the posterior part of the brain, instead of having TPJ and MT, we had 2 regions in the interparietal sulcus. So the conclusion of this fMRI study is, first of all, that not only the cerebellum is involved in saccadic adaptation, but also different areas in the cerebral cortex. And these areas are located in the hemisphere contralateral to the adapted saccade. I forgot to mention that we adapted only leftward saccades. And all the hemispheric areas except the frontal one were on the right side or on the left for the cerebellum, so ipsilateral to the adapted saccade. And as I said already, as I showed already, the network for reactive saccade adaptation partly differs from the network involved in voluntary saccade adaptation. So the next study that we performed had two objectives. The first one was to confirm that the parietal cortex is directly involved in voluntary saccade adaptation, and the second one was to better understand its involvement in this type of adaptation. So we used TMS. The questions, as I said already, indicated here. And the third question was to check whether there is any role of this area for reactive saccades. Despite the fact that we couldn't find any functional change in our fMRI study for reactive saccades. So we used the same, so we used fMRI guided TMS. So that means that we used fMRI data in order to put the TMS coil precisely on the PIPS in each of our subjects. And to do that, the fMRI scans was done exactly in the same condition as in the fMRI saccade localizer task described by Girardin previously. And using this information on thanks to a neural navigation system, we could precisely put our stimulating coil so that in order to be able to stimulate PIPS. And we had 4 sessions for each of the 2 experiments. We have 2 experiments, one for reactive saccades and one for voluntary saccades. And in each of these 2 experiments, we had 4 sessions, 3 in which we tested different timings of single pulse TMS stimulation of the parietal cortex. At the beginning, 30 milliseconds or 60 milliseconds after the beginning of the saccades. And the 4th one was a controlled situation in which we stimulated the vertex. So I will first present the results of the voluntary saccade experiment. First, for controversive saccades, we show here the change relative to pre-adaptation baseline of gain, of saccade gain during the adaptation phase and during the post-adaptation phase in all the 4 different sessions. So the vertex session is shown by this gray zone, and the colored curve corresponds to the different timings of PIPS stimulation. And we observed that when we applied this PIPS stimulation at 60 milliseconds after saccade onset, there was a significant reduction of the efficiency of the adaptation. So there was less change of saccade gain over trials. And this difference was maintained in the post-adaptation phase. And the same was also observed for ipsivassive saccades. So to make the story short, we conclude that TMS over the right PIPS at a 60 milliseconds delay interfered with adaptation of both leftward and rightward voluntary saccades. I also forget to mention that we stimulated the right parietal cortex. So controversive means leftward and ipsivassive means rightward. Now, for reactive saccades, we got a completely different pattern of results. For controversive saccades, we have no significant changes of the different curves when compared to the vertex situation, although there was a trend for better adaptation, a larger change of saccadic gain when PIPS was stimulated at 90 milliseconds. This trend turned out to be significant only at the end of the adaptation phase for evasive saccades, but not, however, that this difference was not maintained in the post phase. So we conclude that for reactive saccade adaptation, there was a tendency to reach a better adaptation of reactive saccades when white PIPS was stimulated 90 milliseconds after the onset of saccades. But again, there was no significant after effect, change of after effect. So to conclude this last study, we can say that the PIPS is indeed causally involved in voluntary saccade adaptation, that this involvement is quite specific to voluntary saccades, at least this interference with saccadic adaptation is specific to voluntary saccade, because we observe an opposite trend for reactive saccade adaptation, although barely significant. And also we propose that this facilitation of saccadic adaptation for reactive saccade could be related to a remote effect of parietal stimulation onto the cerebellum. And then we also disclose that the effect on voluntary saccade adaptation when simulating PIPS was observed only for the intermediate timing, that is when we stimulated around 20 milliseconds after saccadic termination. So just when the eyes have landed near the target, and just when the feedback is coming through. So there are two possibilities. Our results might be interpreted as a direct role of parietal cortex in saccadic plasticity, or as a role in error signals processing, in the processing of error signals which are leading to saccadic adaptation. So to modify the model that I presented earlier, we now can remove this question mark here, indicating that indeed there is some changes at the level of the parietal cortex during adaptation of voluntary saccades, as shown by fMRI and TMS studies. We can also propose that there is some pathway involved in reactive saccades, which is modified during reactive saccade adaptation, probably involving TPJ, and also that the areas in the frontal cortex might be involved in both reactive saccades and voluntary saccade adaptations. So the general conclusion, probably you have them already. I will go directly to the second point in which I'm reminding that the adaptation of reactive saccade and of voluntary saccade present several differences as far as the behavioral properties are concerned. They also are supplied by different neural substrates at the level of the cerebellum and also at the level of the cerebral cortex. And one question for future studies, which has been neglected so far, is how error signals are processed for saccadic adaptation. What are the mechanisms which are treating visual feedback in order to induce saccadic adaptation? And the third conclusion is that something that I didn't stress so far, but it's quite interesting to remark that the anatomical dissociation that I pointed out between reactive saccade and voluntary saccade adaptation parallels the known anatomical dissociation related to the two types of attentional control. So this indirectly suggests that there could be some coupling of this motor plasticity process, saccadic adaptation with visuospatial attention. And in this case, we are trying to answer this question now. In this case, it would be very interesting to see whether this coupling could be used in patients in order to propose new rehabilitation tools for visual attention deficits. So I would like to thank all these people from Impact and collaborators from Marseille, Doug Munoz in Canada, and Bertrand Gemari in Paris, and Martin Frantz and Bas Negers in the Netherlands, and all other people from Impact. Thank you very much.
So questions? Many.
Probably a question related to one of your last points. This is a dynamical process, right? The error at the beginning of the adaptation is larger than at the end. At the end, actually, there is no error, relatively. So you could imagine that there is a dynamic you could use to track which area are actually tracking this error and modifying the saccades over the adaptation process. So I was wondering if you have any idea about this. Because you compare, as I understood, you compare adaptation versus no adaptation in the imaging studies. And what about if you look at what areas are actually tracking this change in error along the adaptation process?
Yeah, that's a very, very good point. The strategy that we used in this fMRI experiment was not compatible with what you suggest. But now that we know that we can elicit both signal changes at the level of the cerebral cortex during saccadic adaptation, this is certainly a way to follow. The strategy that we used was instead to use very short duration blocks of adaptation and repeat them as many as we could, as often as we could in a single session in order to boost the board response. But maybe with stronger fields, more sensitive methods, it could be very nice to study the dynamics of the adaptation and of the error declining process that you are referring to, yes.
You said the subject, he doesn't detect that the stimulus has moved, but can he tell that there's, especially early in adaptation, can he tell if you mixed up sort of trials where it moved and it didn't move, could he tell that anything was different between those trials?
Yeah, usually most of our subjects are unaware of the displacement of the target when they are naive. We are no longer naive as experimenters, and we always feel something strange, or we can clearly detect the jumps when we are experienced. And when we, presumably, even a naive subject, when we mixed stationary with jumping target trials, it would be more obvious for them to detect. But having said that, it seems that the fact that we are aware or not aware of this phenomenon does not really change the behavior, at least the behavior of the error reduction process, and even retention, which is supposed to be a direct measure of the sensory motor adaptation, independently of any strategic response that can occur during the adaptation phase. Even this post effect, this after effect, does not seem to be related to the fact that the subjects are aware or not. Now, if we are starting to look at the brain areas involved, that could make a difference, of course. I have to mention that in our fMRI study, all subjects they are aware, but in both cases, in both with the short duration, the short delay, 15 milliseconds, and the long delay. And so that's why we hope that by comparing directly in these two situations, we would get rid of this effect. That's all I can say now.
Given the way the saccadic system maps onto the attentional system, and you mentioned at the end of your talk that this approach of adaptation could be used to correct attentional problems. Have you started doing work in that direction?
We have started to do that. We have not quite ready to do that in patients, except one or two tests like this, very short tests. We have two projects in mind. The first one would be visual deficits in hemianoptic patient. And also the second one, the second application that we have in mind is visual neglect. And in both cases, we have to play with the right protocol in order to be able to induce adaptation in those subjects, which is not always easy because, of course, we have to elicit saccades, for example, in a field which is either unseen or neglected. So it's quite challenging. So we are currently thinking of doing the anti-saccade task, so we can present a visual target in the good field and also a subject to go to the other direction and provide at the end of this anti-saccade narrow signals in order to induce adaptation of these anti-saccades. We have tested that in normal subjects. It works. And we are now analyzing the data of the first patients.
So would it be as simple as if you can get people with a neglected field to make saccades into it, that somehow or other that in itself will drive attention into a field which is neglected and it becomes less neglected?
Yeah,
as simple as that?
That's the idea. That's the idea. Yeah.
Any other questions? Thank you very much.